Hello everyone, myself Mrs. C.S. Nirali, Assistant Professor, Food Chemistry and Nutrition Department, Kekewa College of Food Technology, Saraswati Nagar Campus, Nashik, under course number FCN 357, course title Instrumental Techniques in Food Analysis. Today we will see experiment number 15, title Analysis of Minor Constituent of Foods, under that exercise number 5, Estimation of Total Carotenoids. Coming to introduction part of carotenoids, they are terpenoid C40 compounds widely distributed in plants. They function as accessory pigments in photosynthesis and as coloring matters in flowers and fruits. Their levels are altered during psychological and pathological conditions, especially by viruses. Some of the commonly occurring carotenoids are simple unsaturated hydrocarbons based on lycopene and their oxygenated derivatives known as xanthophylls. Beta carotene is the most common pigment in this group, found in higher plants. A method to extract total carotenoids, them fresh plant materials, and their determination is described below. Principle: the total carotenoids are extracted and portioned in organic solvent, acetone or methanol, on the basis of their solubility. Carotenoids that are found as esters and hydrolyzing during using aqueous 60% kVH. The amount of carotenoids present in samples estimated colorimetrically at 450 nanometers using beta carotene as a standard. Chemical reagents: acetone or methanol, peroxide free ether, ethanol, aqueous 60% kVH, standard beta carotene solution. Procedure: sample extraction, cut the fresh plant material and grind a known amount 2 grams in a mortar with 20 ml of either distilled acetone or methanol. Filter on Buchner funnel through Whatman number 42 filter paper. Repeat the extraction until the tissue is free from pigments. Cool the filtrates and partition thrice with equal volume of peroxide free ether using a separatory funnel. Add water if necessary to produce 2 layers during initial ether extraction. Evaporate the combined ether layers which contain carotenoids under reduced pressure at 35 degrees Celsius in a rotary evaporator or in a hot water bath. Dissolve the residue in minimum quantity of ethanol. Add 60% aqueous kVH at the rate of 1 ml for every 10 ml of the ethanol extracted to saponify. This will remove chlorophylls, lipids, and also cleave the esterified carotenoids. Keep the mixture in the dark in presence of nitrogen or boil it for 5 to 10 minutes or leave it overnight at room temperature. Add equal volume of water and partition twice with ether. Evaporate the combined ether layers as before and dissolve the residue in minimum volume of ethanol. Measure the absorbance of this solution at 450 nanometers and calculate the carotenoid content milligrams per 100 grams in the sample using calibration curve prepared against a high purity beta carotene. Make suitable dilution of the acetone or methanol extract of plant tissue and measure the absorbance at 450 and 670 nanometers in a colorimeter. If A450 is formed, the A440 values using the following formula. Calculation: C is equal to D into V into F into 10 by 2500. Observation: C total volume amount of carotenoids milligrams. D absorbance at 450 nanometers in a 1 centimeter cell. V volume of the original extract in ml. F dilution factor 2500. Average extension coefficient of the pigment. So by this we will be getting the result of the amount of carotenoids present in a sample. Thank you for your patience listening.